Today, I want to talk to you about sterilization. Not the kind where you cut things to prevent reproduction. No, the medical device kind. Medical device sterilization. What works, what doesn't, how it works, how does sterilization actually work, major technologies that are in use today, what are the successes and downsides of each of the methods, and engineering, biology around it, and regulatory reality. So if you enjoy thoughtful, technically grounded conversations about science and technology with a twist of electric discharges, plasmas, then this channel is for you. Hello, my name is Greg Friedman. I have a PhD in bioengineering. I was at university for over a decade, and now I run my own company doing research in electric discharges. And today I want to talk to you about what is sterilization. What does it really mean? It's not really cleaning or disinfection. That's called sanitization. So you sanitize your kitchen to make sure it's kind of clean, like your skin is intact and you're fairly healthy. So even if you eat a couple of bacteria that anyway came from your cats, that should be fairly harmless. When we're talking about sterilization, the goal is to eliminate all viable microorganisms. We want to prevent any kind of disease, even one harmful pathogen. And you know how big they are, right? Like about this. You can't see them. They're microns in size, and even one on a medical implant can do a lot of very expensive, painful, and potentially deadly harm. And this includes bacteria, spores, which is a very dormant form of bacteria, different kinds of viruses, fungi, etc. There's the slime mold that's gaining sci-fi attention, if nothing else, which is another really cool organism I'll be happy to talk to you guys about in a different video. And how sterilization is measured using sterility assurance levels, SALs. So the oldest and greatest sterilization is heat sterilization. Many, many thousands of years ago, our ancestors would put a sword in a fire, get it hot, and then sterilize the wound. That cauterizes it, stops blood from spilling out, and it also kills bacteria. That's perfect for the wound. Today, we use so-called autoclaves. They use frequently combine water steam with high pressure, with high temperature to sterilize. And they work extremely reliably. They're very widely used. And if we can use them, we do. This treatment denatures protein, explodes the membrane. So basically any kind of microorganism can be killed using heat and high pressure. High heat, high pressure mixed with water steam, it's perfect. It's the greatest. Except those of you who worked in the biolab know that many things aren't compatible with high pressure steam autoclaves. Temperature sensitive materials, materials that contain any kind of soldering. A lot of the materials where there is metal and plastic, and especially microelectronics. So while autoclaves, heat, steam, pressure, autoclaves are probably by far the best, they're not used everywhere because they're not really compatible with many things. And actually many devices in your dentist's office are used even though better devices may be available. The ones that are used are the ones that are compatible with the autoclave. So new research in this direction continues, and it is very important. Another way, pretty popular, chemical sterilization using aggressive chemicals to kill pathogens. Ethylene oxide is something that stands out to me. It's really good at killing things. It's wonderful, you take ethylene oxide gas, you put it in a chamber, and it kills everything. What happens when you open the door? So hydrogen peroxide, ethylene oxide, formaldehyde, they all have been at one point or another used for sterilization. The problem, while they're effective, they introduce toxicity concerns, carcinogenicity, mutagenicity. Ethylene oxide, EPA is about to pass regulation to regulate it down below 1 part per million in the sterilization facility. It's a pretty nasty gas. It's really good at what it does, but there are side effects. It is used today for sterilization of medical equipment that contains both plastic and metal parts. That's probably one of the primary uses for ETO. And one of the anecdotes from my life, I was at a conference basically devoted to ETO, and at a lunch, there were a number of executives from medical device manufacturing companies. And true to my nature, I always ask dumb questions. So I said, guys, I have a dumb question. ETO is horrible for all the workers in the facility. Why do you still use it? And the response is, we got rid of it anywhere and everywhere we could. We use it where we must, where there is no other solutions. So scientists among you, if you have better solution than ethylene oxide, let's go. They're our customers. So that's chemical sterilization. There's also radiation. Using gamma radiation or electron beam radiation damages DNA of the microorganism directly, prevents replication, so basically kills everything. Works really well also, but similar to chemical sterilizers, very special protections, attention is required when building a radiation sterilization facility. As you can imagine, worker exposure to radiation is very problematic. Electron beam, very, very high voltage systems. So any kind of malfunction could lead to significant issues with occupational safety and health. So radiation sterilization is good. It's good and it's widely used in the industry, but it has its drawbacks. There is, of course, why wouldn't I mention it? There's plasma sterilization. Plasma makes a lot of reactive species that are very good at attacking microorganisms, causing oxidative and other kinds of damage. And UV produced in plasma also has strong antimicrobial effects. Plasma can run at low temperature, room temperature. There are issues. Again, same problem with metals. You can oxidize bacteria, you can oxidize metals. There have been problems in the industry with different kinds of solder used in medical devices. Sensitive geometries, depending on what kind of plasma you're using, there may be shadowing effects. It's difficult for plasma-generated species to penetrate deep into crevices. Like, for example, I've done some work with duodenoscopes. These are very special type of an endoscope, which is really, really complicated. It's a mixture of plastics, metals, rubbers. It's got really small openings, high aspect ratio. It's a very difficult thing to sterilize. And while plasma can, material compatibility issues become a big issue. And similar to electron beam, although lower voltage, plasma is still a high voltage system, so malfunctions could be problematic to the operator. So why do some methods fail? Well, bacteria, they're organisms. They try to protect themselves. I've seen bacteria develop, literally, visibly seen bacteria develop resistance to ultraviolet radiation. I was very surprised how fast bacteria develop resistance to, for example, 254 nanometer mercury UV. And bacteria are able to build walls for themselves, so-called biofilms. Biofilms are like an ant hills for bacteria, where the organisms, live organisms, live, survive under layers of stuff. So biofilms are great protection for bacteria. Like if you have biofilm growth in your bathtub, like that black stuff in the corners that you can't scrape off and it keeps coming back, well, it could be mold or it could be biofilm. Another problem why some methods fail is medical devices can be very complex geometries, like the duoendoscopes that I mentioned. Complex geometry, very high aspect ratios, so like very small hole and very long. Material compatibility issues that I keep coming back to. And, like in case of plasma, you may have incomplete exposure of the surface. So it's every method needs to be carefully analyzed, both for its ability to disinfect, but also for its effectiveness in disinfecting. And of course, dose and exposure time matter. Sterilization has some probability involved, right? You're trying to get your active chemistry of some sort, find the right bug. And when you're talking about sterilization for NASA, their definition is, if memory serves me right, less than 2 organisms per square meter of surface. So you're looking at something that's thousands of times smaller than a speckle of sand, less than 2 of that per square meter of surface. That's a lot of surface. So you need to be very careful how you're doing the sterilization, that the disinfectant needs to get to the target. There's almost every video I mention dose and time relationship, the dose and dose rate. And engineering is pretty critical here. You need to understand what types of products your technology can sterilize. And when we're talking about medical products, packaging becomes a big issue. Medical device materials vary. You have the packaging materials, polymers, they're easy to degrade. If you're sterilizing your medical device inside of the package needs to allow the sterilant to penetrate. Kind of easy in case of heat. It's radiative, and in case of radiation, but even steam, will it get through the package? Or, ethylene oxide or plasma reactive species, why would they go through the package? And if the package has holes, why wouldn't bacteria go through those holes? So that's everything you need to consider with sterilant penetration. And of course, there's logistics question. So where you sterilize and where it's used. In your dentist's office, they frequently have an autoclave next door. So they autoclave it in a box right there, and then bring and use it right at the table. Versus like Becton and Dickinson making a syringe, they sterilize it upstate New York, truck it over days and days in the Texas heat, and then use it months later. So logistics also play a very important role. And of course, there is regulatory reality. Food and Drug Administration regulates this in the United States. There are other international standards. There are standards for validation and verification. There are biological indicator standards, like how do you test your autoclave that it works when you designed it? And how do you test it that it's been working the 10 years that you've owned it? they're actually really cool solutions. There's like this tape that you put on your package and it turns different color once the autoclave worked correctly. There's a lot of work is being done in this direction. And documentation, traceability, I mean, there is a whole host of regulatory oversight challenges for sterilization technologies, including plasma. So what is the future of sterilization? What should you think about if you're entering this field, especially if you're entering it as a researcher these days? Well, there is a big push. You know, we have a lot of medical devices that have embedded microelectronics. So you want to look into solutions that are low temperature, that are very gentle for metals and soldered components, microelectronics, microchips, and also compatible with plastics. Plasma-based systems are being investigated in this direction for decades. Low pressure and atmospheric pressure plasma systems. There are, just like I mentioned, your dentist's office, sterilizing on location. There are many solutions where on-site sterilization concepts are being investigated. Even like a syringe, you don't need it sterile until right before it's used. So maybe we develop small, energy efficient, fast sterilizers that we use right before the surgery. And of course, there's big push to reduce toxic sterilants. Ethylene oxide is still widely used. It's bad, toxic, corrosive, carcinogenic, mutagenic. Not a very friendly gas, but it's still used because it works and there isn't a good solution. If you come up with a good solution, you change the whole industry, which is pretty cool. You'll save a lot of lives that way with just one little invention. But it's a tough one. Many people are trying. So in closing thoughts, sterilization is an interesting field. It combines biology, chemistry, engineering, regulatory challenges, lots of different things, logistics. Sterilization as a field of study is quite interesting and quite challenging. And no single method works for everything. There isn't like a silver bullet. So you need to look at the whole system design, not just the core, how it works, but how the thing gets transported, what's it packaged in. There's a lot of future innovation that will focus on safer, better, scalable technologies in this direction. Guys, I enjoy making these videos. If you have questions or comments, please post them below this video. If you have interesting ideas for video that I can make for this channel, please post them there too. I'm always happy to do that. Peace. I'll see you in the next one.